long-range and global physical mapping. The elucidation of order of genes and chromosomes influencing both linkage and co-regulation is at the heart of genetic analysis. Indeed, the characterization of any mutation or cloned gene traditionally includes the determination of its position on the chromosomal map. Genetic and physical maps illustrate the arrangement of genes and DNA markers on a chromosome. All physical maps are based on the direct analysis of DNA. Physical maps are divided into short range and long range. Short range mapping is commonly pursued over distances ranging up to 30 kb. Short range mapping is the average size of a gene and also the average size of cloned inserts obtained from cosmid based genomic libraries. Cloned regions of this size can be easily mapped to high resolution. With restriction enzymes and with advances in sequencing technology, it is becoming more common to sequence interesting regions of this length in entirety. Direct long-range physical mapping can be accomplished over mega-base sized regions with the use of rare cutting restriction enzymes together with various methods of gel electrophoresis referred to generically as pulsed field gel electrophoresis or PFGE, which allow the separation and sizing of DNA fragments of 6 megabase or more in length. PFGE mapping studies can be performed directly on genomic DNA followed by southern blot analysis with probes for particular loci. It becomes possible to demonstrate physical linkage whenever probes for two loci detect the same set of large restriction fragments upon sequential hybridization to the same blot. Long range mapping can also be performed with clones obtained from large insert genomic libraries such as those based on the yeast artificial chromosome or YAC cloning factors. Since regions within these clones can be easily isolated for further analysis, in the future long range physical maps consisting of overlapping clones will cover each chromosome in the mouse genome. Short range restriction maps of high resolution will be merged together along each chromosomal length and ultimately perhaps the highest level of mapping resolution will be achieved with whole chromosome DNA sequences. It is possible to derive long range restriction maps spanning genomic regions that have yet to be cloned. The main utility of such restriction map is to place lower and upper limits on the physical distance that separate two or more DNA markers. Long range restriction mapping requires two tools. The first is a method for separating very large DNA fragments based on size differences, and the second is a set of reagents for cutting DNA at relatively rare restriction sites. The required methodology was invented by Schwartz and Cantor and is known as pulsed field gel electrophoresis. PFGE permits the physical separation of DNA molecules that vary in size up to 9 megabase. The separation achieved is determined by the conditions of electrophoresis. At the lower end, one can obtain separation in the range of 20 to 200 kilobase. Just beyond that possible with classical electrophoresis. At the upper end, one can obtain separation in the range of 1.4 to 9 megabase. Long range restriction maps are best generated by a combination of two approaches. First, single or double digests can be performed on very high molecular weight genomic DNA with a panel of rare cutting enzymes. Second, the same DNA sample can be treated with individual rare cutting enzymes under conditions where partial digestion will occur. All of these samples are loaded into adjacent lanes on the same gel, which is run according to the PFGE protocol, blotted and then probed sequentially with various markers from the region of interest. The physical distance between two markers can be determined by identifying and sizing those restriction fragments or partially digested fragments that hybridize to both markers or only one marker or the other. Prior to the development and easy availability of large insert genomic libraries, the rare cutting enzyme or PFGE approach provided the most feasible means for estimating physical distances between linked loci that are separated by hundreds of kilobases or more. However, it is now often the case that physical mapping is more readily accomplished in the context of clones. Nevertheless, there are still many situations where a region of interest is flanked by two markers that are too distant from each other to allow rapid cloning between them. Genomic restriction mapping can play a unique role in these situations.